Okay, well, hello everybody, and thanks for joining me for this presentation. My name is Russell Coleman. I am Director of Strain Engineering at Primrose Bio Inc. And in this presentation, I'm going to provide an overview of our Pseudomonas-based protein expression technology, the Pfenex Expression Technology, and review how it has been developed to overcome the challenges of difficult to express proteins, and also provide some information and case studies on our latest platform offering, Pfast Protein Expression, which is designed to accelerate therapeutic protein screening. So Primrose Bio is located in sunny San Diego, California, and was founded in 2023 following the merger of Primordial Genetics and Ligand Pharmaceuticals' Pelican Pfenex division. Through this merger, our mission is to create custom manufacturing solutions for next-generation biotherapeutics, encompassing both protein-based and nucleic acid-based therapies. Our two key technology offerings include Pfenex Expression Technology, the focus of this talk, and Prima RNA poles, which are developed to produce mRNA products with improved performance at reduced cost. We also offer Pellicrim 197 vaccine carrier protein, the only commercially recombinant CRIM 197 used in marketed vaccines, and our function generator technology, which improves microbial host performance through genome diversification. We have a modern facility in San Diego, California that enables scaling from initial clone to GMP transfer. Our labs are ISO certified facilities and include broadly applicable purification platforms and advanced analytical tools for protein analysis. With this slide, let me introduce the Pfenex Expression Technology, which is powered by the Pseudomonas fluorescens bacterium and an expression toolbox based on its biology. This technology was specifically developed to deliver cost-effective and high-yield protein production of challenging to express non-glycosylated therapeutic proteins. Pseudomonas fluorescens is a gram-negative, non-pathogenic bacteria found really everywhere in nature, including soil, water, and on plants. In fact, our isolate from which we developed this platform was plucked from a lettuce leaf from a farm here in San Diego, California. And through a combination of informatics and functional analyses over 20 plus years, we have learned how to leverage this organism's natural adaptability and metabolic versatility to build out our expression platform. As a gram-negative bacteria, Pseudomonas fluorescens has all the same advantages with regards to recombinant protein production as another well-utilized gram-negative bacteria, E. coli, since Pseudomonas, like E. coli, has a short cultivation time, simple defined, inexpensive media, and great scalability. However, Pseudomonas has a key advantage in that it is an obligate aerobe. This means it does not accumulate inhibitory organic acids such as acetate during a fermentation process. It simply does not have the machinery to switch over into anaerobic respiration like E. coli can when oxygen becomes limiting in a fermentation. This allows Pseudomonas to generate very high cell densities and high protein yields in an optimized fermentation process. Other important attributes: all the media we use is animal origin free, and no antibiotics are used in our system, both important safety features valued by the regulators. However, it's the last bullet point which describes where this platform really excels: rapid development of optimized and robust expression strains through combinatorial screening. Through this approach, use of Pseudomonas fluorescens system in combination with high throughput expression toolbox screening, this platform has delivered 6 and counting approved therapeutics and vaccines, further validating this platform. This slide shows a high-level overview of the Pfenex Expression Toolbox. The Pfenex technology leverages an extensive toolbox of genetic elements incorporated into our expression vectors and modified host strains, all based on Pseudomonas fluorescens biology. Our compatible expression vectors, shown here at the top right. Many of our strains harbor more than one plasmid, contain different combinations of transcriptional promoters, ribosome binding sites, or translation initiation strengths, secretion leaders, or signal peptides for localizing target proteins into the periplasmic space of cells, often to promote improved folding, particularly with proteins that have multiple disulfide bonds, and native Pseudomonas fusion partners to help promote expression of very small peptides or small proteins. Down below on the right our toolbox also contains modified host strains, including strains that overexpress folding helpers, such as chaperones, disulfide bond formation factors, secretion helpers to help localize that protein into the periplasmic space of cells as needed, and protease deficient strains. Our protease deficient strains, we have single knockouts and stacked multiple knockouts. And many of our strains have a combination of protease deficiencies, but also overexpressed folding helpers. So you can see by combining these elements, you can generate hundreds to thousands of expression strains, each representing a unique combination of toolbox components that can be screened in parallel using our high throughput automation enabled workflows to identify highly productive strains. I'll describe how our high throughput screening workflows operate in a minute, but first let me highlight just one Pfenex Expression Toolbox component, secretion leaders, my favorite component, to show you how screening multiple leaders can have a significant impact on therapeutic protein quantity and quality. Secretion leaders, also known as signal peptides, or short amino terminal sequences, typically 2 to 3 kilodaltons, that signal the cells to translocate proteins from the cytoplasm into the periplasmic space using the SEC pathway. The periplasm is the space between the inner and outer membrane of gram-negative bacteria, and it can be advantageous to localize target proteins within this space, particularly for proteins with multiple disulfides and complex disulfide bond patterns, since it is a less reducing environment. There are native factors there that promote disulfide bond formation. There are fewer and different proteases, and it's one membrane closer to purification. As the protein is translocated from the cytoplasm into the periplasm, the secretion leader is removed. Secretion leaders vary in size and sequence. What leader is optimal for any given recombinant target protein is difficult to predict. So it is best to expression test multiple secretion leaders to assess the quantity and quality of the expressed target protein. Here you see an SDS CGE gel-like image analysis of therapeutic enzyme expression at 96-well scale, evaluating 39 different Pfenex Toolbox secretion leaders. CGE, capillary gel electrophoresis, is very similar to PAGE in that it separates proteins based on molecular weight, only it's more high throughput. Expression was tested in 2 different host strains to provide biological replicates. So we took induced 96-well culture, generate lysates, and ran the lysates on SDS-CGE and looked for an induced band. So let me bring up my pointer to highlight a few things. So in this first lane, we see lysate from cultures that express the recombinant enzyme without a secretion leader. So this is cytoplasmically produced enzyme. So you can see there is a low-level amount of expression. However, comparing that to expression tested using 39 different secretion leaders, really the expression varies. For some secretion leaders, we see almost no recombinant enzyme produced. For others, we see plus and minus the secretion leader, which is obviously not optimal. And for some, we see high levels of expressed recombinant target with that secretion leader removed. And in fact, secretion leader 7 produced or achieved the highest titers at 2 grams per liter of leader removed mature therapeutic enzyme. So hopefully, this illustrates how combinatorial screening of just one toolbox component can significantly impact expression quantity and quality of the target protein. And our fast feasibility case studies will further support this approach. Here is a high-level overview of our high-throughput growth and expression workflows. Typically, our strain building, sample processing, enrichment, or chromatography and analysis occurs at 96-well scale. For the strain building, we will combine our expression plasmids with our toolbox host strains, array them in 96-well plates, perform 96-well electroporation, take those transformants and use them to inoculate these 96-well deep well growth plates, typically half ml culture per well. We'll take the inoculated plates, whip them around pretty good in our Schinker incubators for about 24 hours, and then induce for 24 hours, and then harvest the cultures. So one thing I want to point out, and this is a big advantage of our platform, is typical ODs achieved are in the 30 to 50 range. Again, this is taking advantage of Pseudomonas fluorescens obligate arrow of nature to produce very high biomass and allows us to generate sufficient quantities of protein at this scale to make a very reasonable assessment of both protein quantity and quality produced by each unique strain. You would be lucky, for instance, to achieve ODs in the 3 to 5 range with E. coli. For sample processing, we will take a sample of those cultures right on our automation platform, generate cell lysates or culture lysates by sonication, fractionate those lysates into soluble and insoluble fractions, and analyze each of those fractions for protein production. If a protein has a enrichment tag, such as a His tag, we can perform 96-well high throughput IMAC enrichment. If it's an antibody derivative, we can perform high throughput affinity enrichment. For the analysis, we want to evaluate those culture lysate fractions for the amount of protein produced. As we discussed on a previous slide, our workhorse method for quantification of protein lysates is capillary gel electrophoresis, very high throughput. If a protein has a binding partner, we can assess activity or quantities of actively binding protein from each strain by ELISA or biolayer interferometry. If it's an enzyme, we can develop high throughput enzymatic assays and also cell-based activities as appropriate. So once we acquire the analytical results from a high throughput expression workflow, we can now explore the expression space. We are looking to understand what combination of host strains and expression plasmids are driving the highest titers of soluble and active protein. Once the high producers are identified, they are further evaluated under more controlled high cell density growth conditions at 2-liter scale. Here you see a picture of our 2-liter DASKEEP units, 1-liter working volume. Each strain is evaluated under multiple induction conditions under a DOE approach. Factors include temperature at induction, pH at induction, inducer amount, biomass at induction, for example. The goal here is to quickly identify highly productive strains that are robust under multiple conditions from which to develop a manufacturing process. So utilizing that combinatorial screening approach based on Pseudomonas biology, as we just outlined, the Pfenex technology has been very successful in expressing complex and challenging proteins such as antibody derivatives, vaccine antigens, therapeutic enzymes, immunocytokines, for example, that have previously resisted production in other systems, including yeast, E. coli, CHO, and others. In our system, we've often see soluble and active yields improve 10 to 20-fold. This success rate highlights the Pfenex platform's effectiveness in overcoming production challenges. Having developed multiple approved therapeutic products in our system, we know it's important to develop not only productive strains, but strains suitable for developing a robust manufacturing process. Now that you've had the opportunity to understand how the Pfenex Expression Technology works and how it addresses the challenges of therapeutic protein production, let me introduce our latest platform offering, Pfast. Pfast is accelerated protein expression feasibility in just 10 days. Starting at $25,000, our Pfast program provides new insights into your protein's potential using the Pfenex platform. Pfast provides a low-cost opportunity to really kick the tires on the Pfenex Expression Technology platform without committing to a full-scale development program. A full comprehensive Pfenex platform screen takes typically 9 to 12 weeks to deliver an optimized expression strain ready for cell banking and manufacturing process development. Comprehensive platform screening is a great solution to quickly deliver a robust manufacturing strain to support material generation, often grams of protein for preclinical and IND-enabling studies. However, many of our partners are just looking for quick feedback on expression for a novel protein format or modality, or feedback on a protein that they have seen has been problematic in other systems. To address these early R&D challenges, we offer the Pfast feasibility workflow. So here's how the Pfast feasibility workflow is conducted in just 10 days. In 1 to 2 days, we'll take that synthesized gene, which also encodes a 6X his tag, ligate it into a pool of Pfenex expression vectors, typically 40 to 200, and then transform those pools into 1 to 2 toolbox host strains. We'll then plate to single colonies from those pools. From the transformants, we'll also generate overnight cultures, prep plasmid DNA, and run an NGS QC to confirm that all the intended expression plasmids are represented in those pools. We'll then pick from the single colonies within 3 to 6 days, inoculate wells within those 96 deep well plates, run those growth and expressions in the shaker incubators. 7 to 8 days in, we will generate lysates from those expressions, run CG and or LISA on those lysate fractions, and then ID those highly productive wells for IMAC purification. We then run high throughput IMAC purification, analyze those enrichments by CGE for concentration and purity. And then remember, we don't know with the combination of toolbox components in each well, since this is a pool cloning strategy. But those best hits we can sequence as needed. But the idea here is to generate output on a potential titer. So what are the highest titers generated in those pools? And also we can deliver some potential material, anything left over after analytical assessments, to our partners for their own evaluations. So let's run through a couple of quick case studies. So our first case study is a fast expression optimization for a novel antibody format, Fab-VHH. So for the Fab-VHH fast feasibility, and a representation of the Fab-VHH is shown in the bottom right-hand corner. So the Fab-VHH is a bispecific. It has an antigen binding region, of course, within the variable region of the Fab, and also an antigen binding region in the VHH, which is fused to the CH1. To expression test this molecule, we generated 2 coding sequences, both codon optimized for expression in P fluorescence. So over the years, learning how to express Fabs, we understand that it's important to evaluate orientation of heavy chain to light chain expression with respect to a single promoter. So we synthesized 2 operons where the heavy chain and light chain are co-transcribed, switching the orientation, heavy chain first and then light chain first. We then ligate these synthesized coding sequences into 40 toolbox expression vectors. This will allow us to evaluate whichever coding sequence, the light chain or the heavy chain VHH, is first, with our genetic elements incorporated into our expression toolbox. So in this case, it's a single, very strong TAC promoter, which is IPTG inducible. We'll assess 2 different ribosome binding sites or translation initiation strengths, high and medium, and then 23 different toolbox secretion leaders. So a Fab VHH obviously has multiple disulfide bonds. So it's obviously beneficial to localize this protein into that periplasmic space where the disulfide bond folding can occur. So for that second coding region on the codon optimized genes, we will fix the secretion leader for whichever coding sequence is second on the operon. So we've chosen the secretion leader that we've had very good success with for these type of Fab molecules in the past. So we'll lock that second secretion leader. But the important thing is we'll understand how these toolbox elements impact both the heavy chain VHH and the light chain individually when expressed from these toolbox expression vectors. We'll perform that pool ligation, transform into a single host, and run our expressions. So quickly running through the analysis workflow, we'll perform the fast high throughput expression at 96 well scale. We'll harvest the cultures, take a sample of those cultures, dilute it in PBS 1 to 3, generate lysates from those dilutions by sonication, and then fractionate those lysates into insoluble and soluble fractions by centrifugation. Then we will then analyze those lysates by SDS-CGE. So this is quantitative and qualitative. We're looking for an induced band of soluble Fab VHH. We'll also be looking at the qualitative expression to make sure that the protein, both the light chain and the heavy chain, is intact and not degraded. We'll perform ELISA to assess active binding of the Fab VHH generated by each strain, and then intact mass to ensure that the protein is intact. Those secretion leaders are being processed off correctly as the protein is localized in the periplasmic space, and the protein is fully intact. There's no nibbling due to proteases. Here we see fast CG analysis on the two orientations, the heavy chain VHH first and the light chain first. We were able to obtain some reference material for the Fab VHH and spike that into our lysate that's shown on the far right-hand CGE image. So that way we understand where the heavy chain VHH and the light chain migrate. And then looking at the two CGE images of representative samples for both orientations, on the left-hand side, the heavy chain first, in the middle panel, the light chain first. We are able to identify, those induced protein bands and then quantify that expression based on the CG internal ladder. So here is a CG analysis plotting each isolate in the Pfast screen, looking at light chain by CGE versus heavy chain VHH by CGE in each one of the panels. We're looking at both the orientation, heavy chain first versus the light chain first in the second panel. So again, each dot represents an isolate from the Pfast screening. We don't know the composition of the toolbox components in each one of those isolates. We have a null control run in this expression. So those are represented by the red dots. So this is just empty plasmid, no gene cloned. So we wouldn't expect to see expression in those constructs. And as you can see, it's pretty obvious from this data set that the light chain first orientation did the best job in promoting high expression of both the light chain and the heavy chain VHH. In fact, the light chain orientation promoted expression up to 570 micrograms per mil. We then selected our top 96 strains, 48 from each of the orientation. Those are represented by the empty diamonds, and ran NGS sequencing confirmation to understand what are the secretion leaders and RBS combinations that are driving expression, high expression for each one of these orientations. Once the NGS identified unique expression strategies, so many of these expression strategies are represented multiple times, of course, in these top hits, but we ran intact mass analysis on unique expression strategies identified by the NGS analysis. And those are represented in the green diamonds. So in addition to plotting the CG analysis, we also wanted to evaluate Fab antigen binding by indirect ELISA. So we generated a ELISA standard curve using that Fab VHH reference material, and then used the standard curve to quantify quantities of actively binding Fab in these fast expression samples, quantified in micrograms per mil by ELISA, and then plotted those versus heavy chain VHH quantified by a CGE. So you can see looking at these two different orientations, heavy chain first versus light chain first, the ELISA lines up with what we saw with CGE. Light chain first promotes the highest expression activity. We are reaching 360 micrograms per mil with a light chain first orientation. Since this is a single dilution factor, Some of the samples reached the highest standard curve point at the top of the curve, but we're not looking for individual strains. We're looking to understand which toolbox components are promoting high expression or high active expression of the Fab. Okay, we then ran intact mass analysis on our samples that are promoting the highest expression of soluble and active Fab VHH for both orientations, heavy chain first and light chain first. So that was 21 samples in total, all confirmed to be unique expression strategies by NGS. So we took soluble lysate samples, shot them through an LC affinity column that's directly coupled to an Orbitrap MS to enrich for antibody prior to the analysis. So looking at examples from 2 strains shown in the deconvoluted MS spectra shown on the left. For strain number 1, we were able to detect fully assembled full-length Fab VHH antibody for both the heavy chain first and the light chain first orientations. Right, so to summarize the Fab VHH expression optimization using Pfast, Pfast delivered up to 500 micrograms per mil of soluble and active intact Fab VHH within 10 days for further development. So the best Pfast toolbox combinations identified in the screening, including the structure of the coding sequence. So we understand that the light chain first orientation promoted the best expression. These genes were codon optimized for expression in P fluorescence. We understand the transcriptional promoter required the translation initiation strength and secretion layer combinations that promoted the Fab VHH expression. These expression toolbox identified can enable high titer quality expression for variant library screening based on this antibody format moving forward. But also the Pfast identified expression strains can certainly be scaled immediately into 2-liter fermentation scouting to produce multiple grams of enriched Fab VHH within 5 weeks. So lots of options here moving forward based on the Pfast screening. And again, this was delivered within 10 days. Our second case study is an example of lead identification to support and streamline computational drug discovery. Now let's talk about how Pfast screening can enable lead generation. Lead discovery, a crucial step in drug development, relies heavily on computational design to identify promising molecules that can effectively target a specific disease. Pfast can streamline this approach by providing rapid expression feedback. In the first step, Pfast screening, a model antibody scaffold or diverse variants generated by computational design, let's say a handful, 3 to 5, can be Pfast screened to identify an expression tool subset that enables high titer and quality expression. In the second step, lead selection, computational modifications can then be cloned into selected Pfast tools. We're talking hundreds to thousands of variants to assess expression and activity. Leads can then be enriched for further characterizations. Once leads have been selected, they can be scaled into 2-liter fermentation scouting for mg-scale purification and analysis. Or additional variants can be generated for an additional round of variant screening to dial in on desired properties. Here's a case study where Pfast was applied for lead generation. So a partner generated computational Fab predictions to assess antigen binding for a humanized Fab molecule. In the first step, Pfast screening was conducted on representative Fab computational designs to identify common toolbox components promoting high expression. Coding sequences were synthesized for 3 diverse Fab designs. We ran the Pfast expression, and the goal here is to identify coding sequence designs, secretion later RBS combos effective across all those 3 variants. This is identifying the minimal toolbox for lead screening. In the second step, lead identification, 48 Fab computational designs generated by the partner were screened to identify high expression with improved antigen binding. We used Pfast minimal toolset identified in step 1 And the goal here was to identify 8 candidates for rapid generation of putative leads. So in step 1 of the Pfast screening, and remember the goal here is to identify expression tools for subsequent lead screening, we design 2 operons varying the heavy chain, light chain coding sequence order with respect to the promoter. We 2 operon designs for 3 representative Fab sequences and clone those into 40 expression toolbox vectors, varying the RBS and secretion leader combination. So similar to the first case study, we lock the secretion leader for the coding sequence on the second half of that operon. And using the expression vectors, we will screen the RBS secretion leaders for just the whichever gene, heavy chain or light chain, is on the front end of that operon. So we performed the expression in a single host strain, generated samples, and performed the analysis. Reviewing the analysis workflow for this case study, how we process the samples, how we analyze the samples. We run the high throughput 96-well expressions, sample from those cultures, generate lysates, fractionate the lysates by centrifugation into soluble and insoluble lysate fractions, and then analyze the soluble fraction for Fab production. So in step 1, the goal here again is to identify a subset of expression tools promoting Fab expression. We will take the lysate soluble fractions, analyze them by capillary gel electrophoresis under non-reducing conditions to assess assembled Fab production. In step 2, we take those subset of productive expression tools and then screen computational variant designs for the Fabs. Assess those lysates by, again, capillary gel electrophoresis, and then also measure the lysates for actively binding Fab by ELISA. And the goal here in step 2 is to identify the computational design impact on Fab expression. So in the step 1 CGE analysis, we were able to detect soluble expression of up to 494 micrograms per mil of only for the Fab variant 1 design. So the soluble lysate fractions were analyzed by CGE. So an example of gel-like image for Fab variant 1 is shown above for the 2 orientations, heavy chain first in the operon versus light chain first in the operon. So we're looking to detect and quantify an induced band at approximately 50 kD, representing assembled Fab, is shown by the green arrow here. And plotting a soluble Fab titer in micrograms per mil versus the 3 Fab variants, variant 1, 2, and 3, each with their different orientation, heavy chain first, light chain first. The results are quite dramatic. We saw very high expression only from the Fab version 1 and only from a single orientation, heavy chain first in that operon. Again, just a reminder, each dot represents a single isolate, and the red dots represent our null strains. So these are strains that do not have a Fab gene cloned. Continuing with the step 1 analysis, we are looking here to identify vectors driving high Fab version 1 expression based on CGE. Again, we only saw Fab version 1 and the heavy chain, light chain orientation really promoting high Fab expression. So 24 of the highest Fab version 1 production strains shown in the green diamonds in the plot were selected for vector identification by NGS. What are those toolbox components in the vectors for this highest expression strains? NGS identified our 5 most highly represented toolbox plasmid strategies. Those are shown in the table above. And vectors Pfast 201 or 021 and Pfast 041 were identified at the highest frequency by NGS. Pfast 021 utilizes the TAC promoter medium RBS leader 3 to drive a Fab version 1 titers, and Fast 41 uses TAC RBS high in combination with leader 11. So these are the two vectors that will be used in step 2 to expression screen the different Fab variants. In step 2, we performed the Fab lead identification expression testing. The partner provided 48 computational designs, all based on Fab version 1, with that heavy chain first in that operon orientation. And we generated or synthesized genes for expression testing in this step 2. So that's 48 variants, computational designs, times 2 expression vectors that were identified in step 1, Fast21 and Fast41, times 3 different host strains, toolbox expression host strains. So that equals 288 unique expression strains that were grown in duplicate. So in this step 2, we do not use a pooled cloning strategy. So we do know or understand the plasmid host strain combination in each well in the 96 well expression test. So we looked at 3 different host strains as mentioned. The first host strain is just our wild type, or our least modified toolbox strain. A second host is a strain that promotes paraplasmic localization of the Fab, and host strain 3 overexpresses disulfide bond promotion factors for improved folding in that paraplasmic space. In the step 2 CGE analysis, we are looking to quantify assembled Fab, about 50 kilodaltons, looking for this induced band, again in this CGE example. So we plotted each of the Fab variants versus the 3 host strains versus the 2 expression plasmid strategies identified in step 1. So comparing the expression to in the diamond here, you see the line. This is the step 1 Fab version 1 expression strain. Many of the variants actually exceeded expression well above 1,000 micrograms per mil, while other of the computational variants fell well below that line. However, it was RBS medium, leader 11, and host strains number 1 and 2 that promoted soluble Fab expression, greater than that 1,000 micrograms per mil. In step 2, we also ran LISA analysis on those expressions, measuring the Fab binding to an antigen. So the fold change by ELISA is shown in this top panel and compared to the Fab expression by CGE, which was shown in the previous slide. So again, the line shows the results for the step 1 Fab version 1 expression strain as a control. And many of the variants seem to produce higher antigen binding as well as expression, particularly for when combined with host strain 2 using RBS medium and leader 11 on that heavy chain. So the partner can take back the ELISA and the CGE data. This data can inform additional rounds of computational design and expression testing using Fab. Also, in this case, the partner also wanted to select specific Fab variants. Those are highlighted in green for mixed scale affinity purification at 96 well scale. So this is the generate Fab material for further characterization. So within 8 days, we completed this step 2 expression test, completing the screening, delivering the data to our partner, who will then use this information to identify potential lead Fab candidates. Okay, so hopefully these 2 brief case studies illustrated how Pfast can be utilized to optimize expression for novel antibody formats, as well as enable rapid expression feedback for computational designs. So Pfast, again, is rapid expression feasibility in just 10 days. The program deliverables include a detailed report providing titer analysis of soluble and insoluble lysate fractions by SDS-CGE and or ELISA. Optional analytical characterizations can be provided, including those assays listed there. And if any sample material is available after analysis, we can provide that to our partner. After the Pfast evaluation has completed, multiple options are available to continue leveraging the Pfenex platform, including scale-up, further strain optimization, and of course, licensing of the strains. So to learn more, please visit us at our website, www.primrosebio.com/fast. All right, well, thanks for joining me for a quick Q&A. So I've listed 3 questions that we typically are asked about the Pfenex Expression Technology. This first question is, what types of proteins can be expressed using FAST? Are there any proteins that are not appropriate for FAST evaluation? I would answer that, we've seen all classes of protein through 20 plus years, including antibody fragments and derivatives, next generation antibodies, cytokines, vaccine antigens, research tool proteins and enzymes, and other very complex disulfide bonded proteins. The Pfenex Expression Technology has been up to the challenge. However, glycosylation does not happen in a microbial or bacterial system. So if a protein requires glycosylation for folding and activity, that's probably not appropriate for the Pfenex Expression Technology or in any bacterial expression technology. So another question I'm often asked, question number 2, is why is protein produced in Pfenex of higher quality compared to other systems? And the answer is screening our toolbox, right? Parallel combinatorial screening of our toolbox components identify the optimal expression strategy for any therapeutic protein the first time through, right? There's no iterative screening. So we screen through the toolbox and essentially let the biology tell us what is the best strategy for any given recombinant protein. So a reminder, the toolbox components screened using Pfenex are the genetic elements incorporated into our expression plasmids. We also screen protease knockout strains, strains that overexpress folding helpers, strains that overexpress disulfide bond formation factors, and strains that promote paraplasmic localization. So, as I mentioned in the previous question, this is a bacterial system, so we do not make glycosylated proteins, so that can be advantageous to avoid any heterogeneity issues that are often seen in other systems such as yeast. And the last question that I'm often asked, are there any production or analytical issues associated with Pfenex or Pseudomonas fluorescens bacterium in general? The answer is no. Fermentation, primary recovery, and downstream purification processes can all be performed using standard equipment used for production of therapeutic proteins. And any E. coli process can be quickly adapted for use with Pseudomonas fluorescens, no problem. Also, in addition, analytical methods used in characterization and testing proteins produced in other microbial systems can be used to characterize products produced in Pfenex. Additionally, a host cell protein assay kit specific to Pseudomonas fluorescens is available. So no production or analytical issues. We developed processes for therapeutic production in Pseudomonas fluorescens and have transferred these processes to CDMOs all over the world very successfully. Okay, thank you for your time and attention for this presentation. Again, my name is Russell Coleman, Director of Strain Engineering at Primrose Bio. You see my email there. If you have any questions after viewing this presentation, feel free to shoot me an email. I'll be happy to answer any questions. If you want to scan the QR code up in the right-hand corner, that'll take you directly to our website focused on fast feasibility. Again, thank you.